In this video, we are going to study about teratology or embryopathy. So it is defined as the pathology of the unborn. It deals with the study of the cause and the nature of the congenital anomalies which range from gross structural defects to anomalies at cellular and molecular level. All congenital defects may not be detected at birth. Some are expressed as age advances. Example, congenital dislocation of the hip, patent ductus arteriosus. The normal development is an integrated process of cellular proliferation, differentiation and growth. In this complex process, a series of organizing centers appear in succession in the embryonic area and elaborate inducing substance which differentiate the various tissues and organs in proper sequence. The chemical nature of the inducing substance, which is called as evocator, is not properly known. The overall control of the entire process is performed by the genetic blueprint of the zygote, and this is assisted by the environment, both endogenous and exogenous. Any deviation from these normal performance lead to the congenital malformation. Severe form of defects, which is known as monstrosities, produces abortions or fetal death. Thereby, they are eliminated from the population by natural selection. Sublethal form of defects are expressed as abnormal phenotype. It is probable that 2 to 3% of all live-born infants show one or more significant congenital malformations at birth. It is estimated that about 10% of the abnormalities are caused by genetical and chromosomal disorders and 10% due to environmental factors, and the rest are produced by the complex interaction of both genes and the environment. The causes of teratology falls under two major groups. One is genetical factor, the other one is the environmental factor. First, we'll see about the genetical factor. The genetical factors include the disorders due to the chromosomal abnormalities and the disorders due to the mutation of the gene. Next, we'll see the disorders due to the chromosomal abnormalities which affects the autosome. So, you have the conditions called Cri-du-Chat syndrome, which is due to the 5p minus, that is p is the short term of the chromosome of 5 and minus is deletion. So deletion of the short term of the 5th chromosome leads to Cri-du-Chat syndrome. Next is the Pata syndrome due to the trisomy 13. So you have an extra chromosome of 13. Next is the Edwards syndrome, which is due to the trisomy 18. So you have extra chromosome of 18. Then you have Down syndrome due to the trisomy of 21 or translocation between D and G 21, 22, 22 and 21. Next you have chronic myeloid leukemia, which is due to the translocation of 9, 22 of long, Q is the long arm of the 9 and 22, 3, 4 and Q 1, 1. Next we'll see the disorders due to the chromosomal abnormalities affecting the sex chromosome. So first is the Klinefelter syndrome, which is due to the one extra X chromosome, XXY or XXXY or XXY, or you have quadruple X and Y. Next you have Turner syndrome, which is due to a deletion of X chromosome. So you have total number of chromosome will be 45 with the sex chromosome X and O, or XX bar XO or XXQI. That is, Q is the long arm, I is the isochromosome. Next, you have triple X syndrome, where you have the sex chromosome X of 3 in number. Next, you have XYY syndrome. Next, we'll see the disorders due to the mutation of the genes, which includes the autosomal dominant trait. Under this, you have achondroplasia, angioneurotic edema, familial hypercholesteremia, Huntington's chorea, Marfan syndrome, multiple neurofibromatosis, nail patella syndrome, osteogenesis imperfecta, syndactyly and polydactyly. So these are autosomal dominant traits. Next, we'll see the disorders due to the mutation of the genes, which includes the autosomal recessive traits. So under this, you have inborn errors of metabolism and hemoglobinopathies. Under the inborn errors of the metabolism, we have albinism, alkaptonuria, galactosemia, phenylketonuria and Tay-Sachs disease. Under hemoglobinopathies, you have sickle cell anemia, thalassemia major in homozygotes and thalassemia minor in heterozygotes. Next, we'll see the disorders due to the X-linked recessive traits, which includes the hemophilia, partial color blindness, Duchenne's muscular dystrophy, G6PD hydrogenase deficiency and testicular feminization. Next, we'll see the environmental factors. The mammalian embryos are very sensitive to the influence of various external teratogenic agents. Most of the malformations seen in the human beings have been reproduced experimentally. A proper scrutiny of the environmental factors causing these defects is of immense value, because these factors, unlike genetical hazards, are under human control. The teratogenic factors are classified into 5 major groups. They include physical. Under physical, you have X-rays, radiation, etc. And chemical, the drugs used by the pregnant women. And nutritional, due to hyper or hypovitaminosis, mineral excess or deficiency. Hormonal, due to the maternal diabetes, uses of synthetic progesterones, estrogen and cortisones, etc. And maternal infection, which includes virus, toxoplasmosis, syphilis, etc. So when you see the physical factors, radiation from the X-ray or radium produces skull defects, spinal bifida, blindness, cleft palate and the defects of the extremities. The nature of the malformation depends on the dose of the radiation and the stage of development at which the radiation is given. The studies of the offsprings of the Japanese pregnant women who survived after atomic bomb explosion over Hiroshima Nagasaki revealed that 28% of the surviving children had abnormalities of CNS such as microcephaly and mental retardation. Next, we'll see about the chemical agents. A recent NIH study discovered that about 900 different drugs were taken by pregnant women with an average of 4 per individual. So relatively few of many drugs used during pregnancy have been found to be positively teratogenic for the child. Thalidomide, it is an anti-nauseant and a sleeping pill, may adversely affect the development of the limb buds, which leads to amelia or meromelia. The next drug is aminopterin. It is an anti-metabolite and antagonist of the folic acid. It is used sometimes to induce abortion. When the abortion fails to take place, a malformed child is born showing anencephaly, hydrocephalus, hair lip or cleft palate. The next drug is the diphenylhydantoin. So it is an anticonvulsant drug which is used by the epileptic women, which produces a broad spectrum of abnormalities including the craniofacial defects, nail and digital hypoplasia and mental deficiency, and this is known as the fetal hydantoin syndrome. Prolonged use of diazepam by the pregnant women as a tranquilizer has been demonstrated to increase the cleft lip with or without cleft palate in the offspring. A number of other compounds might be damaging to the embryo or fetus. Most prominent are the thiouracil, which causes goiter and mental retardation, streptomycin, which leads to deafness, sulfonamides, which leads to kernicterus, imipramine, which is an antidepressant which leads to limb deformities, and tetracycline, which leads to bone and tooth anomalies. Intake of aspirins in large doses during pregnancy is potentially harmful to the developing offsprings. In today's society, the use of social drugs like LSD has been reported to produce limb abnormalities and malformations of CNS. Alcohol ingestion by the pregnant women may be associated with short palpebral fissures. Next, you have the hypoplasia of the maxilla, limb deformity, ventricular septal defect, and mental retardation, and these malformations are collectively called as the fetal alcohol syndrome. Next, we'll see about the nutritional factors. Maternal iodine deficiency may cause stillborn or malformed child. Hypervitaminosis A in the later part of the pregnancy damages the cerebral cortex and produces mental retardation. Mercury containing fungicides sprayed over seed corn and food grains when taken by the expectant mother produces birth defects to children with multiple neurological symptoms, and this was reported from Japan, USA and Iraq. Next, we'll see about the hormonal factors. First is the maternal diabetes. Maternal diabetes during pregnancy is associated with abortion or neonatal death or birth of unusually large child. A variety of malformations have been observed, in particular skeletal defects with agenesis of sacral vertebra. Next, synthetic progesterones are frequently used during pregnancy to avoid abortion, and prolonged use of the drug may induce a masculinizing effect in female child, and it is manifested by enlarged clitoris and partial fusion of genital folds. Cortisone, when taken in early pregnancy, may produce a deformed child with cleft palate. Next, we'll see about the maternal infection. The viruses of German measles or rubella affecting the pregnant women in the early stages of gestation may produce various abnormalities of the fetus, such as congenital cataract, deafness, patent ductus arteriosus, septal defects of the heart, dental defects, and mental retardation. Cataracts results from infection during the 6th week, deafness in the 9th week, and cardiac defects between 6th and 9th week. Only 3 viral diseases, rubella, cytomegalovirus, and herpes simplex, have been positively identified as causing malformation and chronic fetal infection which persist after birth. Other viral infections such as measles, chickenpox, mumps, and poliomyelitis may act as a teratogen producing fetal malformations. Most of the above mentioned infections are associated with hyperthermia, which has recently been implicated as a teratogen. A parasitic infection of the expectant mother known as toxoplasmosis has been observed to affect the child, producing hydrocephalus, microphthalmia, and other ocular defects. Syphilis may cause congenital deafness and mental retardation of the offsprings. Next, we'll see about the mode of actions of the teratogenic agents. To understand the actions of the teratogens, certain principles may be laid down. First, the stage of development at which the teratogen act. First 15 days of gestation are usually immune to the action of the teratogen. Most of the teratogen exert their influences during the state of differentiation at the embryonic period, which ranges from 4th to 8th week of development. Each organ passes through a critical period of maximum susceptibility to the action of the teratogen. The chain of malformation depends on the organ or group of organs which are more susceptible at the time of teratogenic action. The same substance can produce different malformation if given at different stages of morphogenesis. When more than one primordium develops simultaneously, the same agent can result in multiple malformations. The teratogen act in a specific way by altering the cellular metabolism. They may interfere with the organizing centers of the embryonic area and elaborate altogether different inductive substance. When two teratogen given at a particular time produces completely different type of malformation, they act on the metabolic process in entirely different manner. Next is the role of genotype on the action of teratogen. An agent teratogenic for one species may not be for another species. When an abnormal hereditary trait appears sporadically in a species, the administration of the teratogen exaggerates the frequency of the incident of that trait. Next we'll see the clinical application. Tumors associated with gastrulation. Remnants of the primitive streak persist sometimes in the sacrococcygeal region, and these clusters of pluripotent cells proliferate and forms the tumor which is known as the sacrococcygeal teratoma, which is commonly contain tissues derived from all the three germ layer, and this is the most common tumor in the newborns.